Welcome to a single-cell multiomics workflow, how to understand the impact of human disease variants on relevant cell states, presented by BioCompare and sponsored by BioLegend. My name is Tamlin Oliver, Managing Editor of BioCompare, and I will be the moderator for today's webinar. David Muench from Eli Lilly is going to talk to us about a broadly applicable multiomics workflow that can be used to determine the impact of human disease variants. He'll get started shortly, but first I want to go over a few procedural details. We'll hold a live question and answer session at the end of David's presentation. You can submit a question for him at any time using the Ask a Question tab in the upper right corner of your screen. The right side of the screen also features an overview of the webinar as well as more information on the speaker. If you have technical difficulties at any time, please click on the Test Your Connection button at the bottom of your screen. From there, you can access webinar support. We've also provided social media widgets beneath the webinar window to facilitate the sharing of this webinar with colleagues. With that done, I can properly introduce David and we can get started. David earned his bachelor's and master's degrees in biomedical engineering at the University of Louisville. He then moved to Cincinnati and worked as a research assistant studying the epigenetics of T-cell memory for a year before becoming a student in the molecular and developmental biology graduate program at Cincinnati Children's Hospital Medical Center. David's thesis work focused on the transcriptional regulation of myeloid development, myelodysplasia, and leukemia. Through his work, David has become an expert in the molecular analysis of transcriptional networks with a special focus on quantitative single-cell techniques. He's currently a postdoctoral research scientist at Eli Lilly, where he's exploring the role of macrophages in autoimmune diseases. Welcome, David. Thanks for joining us today. Let's get this webinar started.
Thank you, Tamlin, for that introduction and for the opportunity to share our work and highlight our single-cell multiomics workflow here. And this work was done in the lab of Lee Grimes during my graduate work. The scientific community as a whole is becoming very good at detecting genetic variants associated with human disease due to advances in sequencing and associated technologies. However, to understand the significance of a given variant, it's important to determine the causality in disease. And to do this, we turn to disease models in which we can make genetic modifications to model these variants. So these include cell lines or even model organisms like mice. However, there are still some significant limitations that impede our understanding of the causality of the detected variants. So these include the need for additional technologies and a lack of proper understanding of normal biology or a normal biological process. So on the technology front, the advent of single-cell RNA-seq has really made a huge impact in the field and powered downstream analyses. And you can see that the number of single-cell data sets has grown exponentially over the past few years. But we're still left with a question of how to make accurate cell state comparisons in this data. So this becomes important when comparing wild type and disease states to understand the impact of disease. This also influences how we can understand therapy and the impact of therapeutics, as well as understanding transitions in disease states from active to remission to relapse. In an effort to understand normal biology, the Grimes Lab has recently published a paper that utilized a new bioinformatics algorithm called ICGS to order single-cell RNA-seq data. The results of this paper highlighted rare transition states in hematopoiesis and identified key lineage decisions between the monocytic and granulocytic lineage. But we're left with a few questions after this study. For example, can this roadmap of normal gene expression be used to better understand disease states? And this brings up an important point that must be addressed, and we asked ourselves, how could we accurately compare the mutant cells to the normal, which, as you can see, is composed of many different cell states. And so we wanted to develop a new technique to address this important problem. As I mentioned, the Grimes Lab uses hematopoiesis as a model system to understand transcriptional regulation of development. And here is shown a schematic of the hierarchy of hematopoiesis, which proceeds from a long-term hematopoietic stem cell through a series of intermediate cell states and finally mature effector cells are formed. And so this hierarchy is currently being challenged in the field, and additional cell states have been identified. But today we'll focus on the development of neutrophils, and there are several stages in the development of neutrophils, including specification or the ability to form neutrophils, commitment or the decision to stick with the neutrophil lineage, and also maturation of the mature neutrophils. The formation of mature neutrophils is controlled by a number of transcription factors, including GFI1. And GFI1 variants have been identified in patients with severe congenital neutropenia, or SCN, which is a rare disease characterized by a significant reduction in the total number of neutrophils in the blood of these patients. And this makes these patients susceptible to recurrent infections. Interestingly, despite the rescue of neutrophil numbers by injecting them with GCSF or granulocyte colony stimulating factor, these patients remain susceptible to infections, even though they have elevated numbers of neutrophils. This suggests a functional defect in these neutrophils, and that's not fully understood. So, as shown in the table, there are a number of GFI1 variants, and the impact or significance of many of these variants is not fully understood. So in this study, I will discuss how we modeled patient-derived mutations in mice to identify 2 defects in granulopoiesis. Then we'll revisit normal steady-state terminal granulopoiesis to identify a new transition state. Then we'll return to the mutant mice for an in-depth multiomics analysis to further dissect these 2 defects that we found. And this leads us to a key finding that highlights the importance of analysis at the cell state level. And then finally, we perform a genetic rescue of the mutants to identify the underlying defect in neutrophil function in these mice. So first, let's talk about our new mouse models of neutropenia. So as I mentioned, there are a number of GFI1 variants that have been identified in neutropenic patients. However, a majority of these have not been functionally assessed. So to validate the impact of these variants, we leveraged findings from Andre Olson's paper in which he identified a gene regulatory network controlling the monocytic and granulocytic lineage decision. And this network is led by GFI1 and IRF8. And notably, IRF8 is a direct target of GFI1, and GFI1 represses IRF8 expression. So we utilized IRF8 GFP mice where GFP is fused to IRF8. So the level of GFP reports IRF8 levels. So we took cells from these mice and transduced them with lentiviral vectors to express the GFI1 variants and looked at the effect on IRF8 GFP levels. So as you can see on the left, wild-type GFI1 represses IRF8 levels as expected. The known mutant P2A deregulates IRF8. However, a majority of the variants do not directly affect IRF8 levels, except for a number that are clustered near zinc finger 5 and zinc finger 6 of the GFI1 protein, including N382S, K403R, and R412X. And so this was interesting to us because zinc finger 6 has been previously shown to be dispensable for GFI1 function in vitro. However, the presence of these mutations in this coding region suggests a functional role for this domain. So we wanted to further analyze these 3 mutants. And so we generated mice with these 3 mutations in the GFI1 locus. And first we checked for the expression of GFI1 protein in hematopoietic cells. And unlike GFI1 knockouts, the 3 new mutants form GFI1 protein, suggesting that these are, in fact, new mouse models to study that do not simply copy the GFI1 knockout. Notably, the R412X mutation is smaller because it's a truncation mutation. However, it is less abundant than the other GFI1 proteins. So we first looked for the presence of neutrophils in the peripheral blood of these mice. So similar to GFI1 heterozygous mice, which have plenty of mature neutrophils marked by CD11b and Ly6G, K403R also has normal numbers. However, the R412X mutant has significantly fewer neutrophils, whereas the N382S mutant lacks peripheral blood neutrophils similar to the knockouts. However, if we fact sort out the neutrophils from these mice and look at their morphology, we note that those isolated from R412X appear a bit abnormal or immature, suggesting a second defect in these mice. Not only production, but perhaps a second defect. Since the N382S mutant mice did not form any neutrophils whatsoever, we decided to focus our analysis on the K403R and R412X mutants. So to investigate the possibility of a functional defect in these mice, we challenged the mice with pathogens that require neutrophils for clearance, C. albicans and Staph aureus. Following infection with C. albicans, the R412X mice succumbed very quickly. However, the K403R mice, which have normal numbers of neutrophils, survived perfectly fine. However, during a Staph aureus infection, both K403R and R412X mice succumbed very quickly. These results suggest the presence of two defects in the mice. The first is production of neutrophils, and the second is the function of neutrophils, which is independent from their production. In an effort to understand the cause of these defects, we remembered that the R412X mutant makes less GFI1 protein. So we hypothesized that this was a hypomorphic mutation. So we bred the mice to homozygosity and found that this partially rescued GFI1 protein levels as well as neutrophil levels. However, those neutrophils still have immature or abnormal morphologies. And challenge of these mice, again with our pathogens, reveals that this partial rescue of neutrophil number was not sufficient to rescue function. Our results identify that patient-derived mutations induce both neutropenia and innate immune effector defects in these neutrophils in vivo. So again, this highlights two separate defects in these mice, the formation and also the function of neutrophils. Before delving into a deep analysis of these mutant mice, we first revisited normal steady-state terminal granulopoiesis to make sure we have a firm understanding of the process. To do so, we first developed a new FACT strategy to analyze terminal myelopoiesis. So we removed lymphoid and erythroid cells from the analysis before gating on granulocyte monocyte progenitors that express Ly6C and CD117 or c-KIT. Then we looked at known markers of granulocytic development, CD11b and Ly6G, as well as monocytic marker CD115. We noted that cells that gain Ly6G do so from a CD11b high state, whereas the monocytic cells gain CD115 from a CD11b low state. And this process is best visualized in 3 dimensions, where you can see that Ly6G positive cells arise at a CD11b high state. And to investigate if cells gaining Ly6G are indeed those that are undergoing terminal granulopoiesis, we FACT sorted cells that are CD11b positive and either Ly6G low or Ly6G high, and looked for changes in nuclear morphology, which is a hallmark of neutrophil development. We found that those cells with high levels of Ly6G expression were enriched in the most mature forms. To further confirm this, we used Fuchi2 transgenic mice, which reports the cell cycle using 2 separate fluorescent proteins. And we find that those cells with high levels of Ly6G are rarely found in cell cycle, further confirming that CD11b positive cells are committing to terminal granulopoiesis. To better understand the transcriptional changes that accompany terminal granulopoiesis, we FACT sorted CD11b positive cells and analyzed them using single-cell RNA-seq. We clustered the newly captured cells with our previous data and identified additional granulocytic clusters. We term these pro-new, pre-new, and im-new, borrowing the nomenclature from the B cell field in an attempt to unify the field. and were encouraged by a recent review article by Laiguan Ning and Andres Hidalgo. And on the bottom, you can see a progressive loss of cell cycle gene expression from pro-new to pre-new, and finally an absence of gene expression in the mNu cluster, suggesting that we have indeed captured the terminal state in granulopoiesis. We further confirmed the ordering of our data by looking at known myeloid genes, shown on the right. For example, IRF8, the monocytic transcription factor, is absent in these newly captured cells. GFI1 expression decreases after the pro-new stage. We can see known transcription factor exchanges, such as the MIC-MAX-MAD-MAX transition and the CBP alpha CEBP epsilon exchange. The neutrophil granule proteins also follow the projected order. Primary granule proteins turn on first, followed by secondary, and finally tertiary granule proteins. We also note a number of differentially expressed cell surface markers, including VCAM1, which exhibits a gene expression pattern similar to GFI1, suggesting that it is marking cells that are undergoing specification of the granulocytic lineage. So in summary, we believe we have captured the cells from the beginning to the end of neutrophil differentiation. To better understand the trajectory of granulocytic development, we decided to utilize CITE-Seq, or cellular indexing of transcriptomes and epitopes. And at the time, it was lucky for us that BioLegend TotalSeq-A platform was just being launched, and they had 4 available markers: c-KIT, Ly6C, CD11V, and Ly6G, which fits perfectly into our analysis plan. And so this technology uses barcode conjugated antibodies that are polyadenylated, similar to endogenous mRNA, so they can be integrated into the 10x Genomics workflow. Since then, BioLegend has built their catalog to include hundreds of antibodies against mouse and human surface markers. They have also developed the TotalSeq-B platform to enable capture using the 10x Genomics 3' feature barcoding, as well as the TotalSeq-C platform, which captures using the 10x Genomics 5' workflow. As you may know, these developments will allow for multiplexing CITE-Seq analyses with other very powerful techniques, including CRISPR-Cas9, perturbations, or immune repertoire profiling. CITE-Seq has many benefits, including the ability to identify cell types better than mRNA-based identification, as shown in the bottom in the PBMC dataset. Another benefit is that CITE-Seq data mirrors FACS data pretty well. So as you can see, FACS data on the top, you can identify T cells and B cells here, and on the bottom, it shows CITE-Seq data showing a similar distribution and expression pattern of cells. So this links the FACS gates to your sequencing data and allows you to ask more powerful questions. For example, as cells proceed from the pre-new to the mNu stage, do they successively gain Ly6G expression? Also, using CITE-Seq, we can identify the transcriptomes of Ly6G high cells. Another aspect of CITE-Seq is cell hashing, which allows you to multiplex samples as well as aid in doublet discrimination. So to perform CITE-Seq, you label your cells with barcode conjugated antibodies, and then this feeds directly into the 10x Genomics workflow, where single cells are encapsulated in oil droplets, and the mRNA and the barcode conjugated antibodies are captured for reverse transcription and library preparation. Then you can pool your mRNA-derived library and your ADT or antibody-derived tag library and multiplex them with other samples for sequencing. So we revisited our novel flow scheme, and shown on the right, we captured a modified GMP gate, which includes all of those cells undergoing both monocytic and granulocytic specification and commitment. And shown below, the clustering of the newly captured cells based off our previous ICGS states show a similar pattern of gene expression. And on the very bottom, we also see similar changes in cell cycle gene expression. And in the middle, you can see a heat map of the ADT counts. So if you focus on the ProNu1 cells, you can see that they express CD117. This expression is progressively lost as cells gain Ly6C, then CD11B, and finally Ly6G expression. So next we utilized a fun tool called SeqGeek to visualize the ADT data similar to how we visualize FACS data. So shown in gray are all cells captured, and those in red are those falling under the individual clusters shown above. So we can see that as cells progress through granulocytic development, they begin at a CD117 high stage, they gain Ly6C, and then slowly drop CD117 expression. Simultaneously, in the middle plots, you see cells begin at a CD11V low stage, then they gain CD11V, followed by Ly6G expression. However, strikingly, cells do not progressively gain levels of Ly6G during development, but in fact, you can find Ly6G high cells in 4 separate cell states, suggesting that Ly6G levels alone are not sufficient to identify these 4 distinct cell states. And on the bottom, if we plot the top 1% of cells expressing the displayed markers, we can link this gene expression to our flow gates. And in summary, the CITE-Seq analysis has provided us with a trajectory of our transcriptional data. Next, we asked if it was useful to perform the CITE-Seq analysis, or if we could simply rely on the gene expression of those genes encoding the surface markers. So we correlated ADT expression to that of the genes encoding the surface markers and found that although they are positively correlated, it's a very weak correlation, highlighting the utility of CITE-Seq. So in a final analysis of terminal granulopoiesis, we identified a new transitional population, pre-Nu1, which bridges the pro-Nu stage of development, where cells are specifying to become neutrophils, and the later pre-Nu and im-Nu stages, where cells are committing to becoming neutrophils. And after using ERD pseudotime analysis to confirm the developmental trajectory of these cell states, we find that the pre-Nu1 state is accompanied by a dramatic loss in gene expression, which includes cell cycle gene expression. However, it's not a complete loss of gene expression because genes encoding functional processes are being launched at this stage. So processes like chemotaxis or neutrophil degranulation are gaining expression, suggesting a switch in function and preparing the neutrophils for their job in the periphery. So by revisiting terminal granulopoiesis, we've identified a new rare transitional state that bridges the specification and commitment stages. So to do this, we utilize novel flow cytometry and CITE-Seq analysis to give a trajectory to our transcriptional data. And this highlighted that our neutrophil effector genes are being launched during the commitment and maturation process. So now that we have a firm understanding of terminal granulopoiesis, we can now revisit our GFI1 mutant mice to better understand the transcriptional deregulation that accompanies the neutropenia and neutrophil functional defects. But first, we're presented with a problem that I mentioned earlier, and that's how can we accurately compare mutant cells to the normal cells, which, as I just described to you, is composed of many different cell states that accompany terminal granulopoiesis. And so to address this, Nathan Salamonis has developed the cell harmony algorithm, which uses predefined ICGS states in our for example, in our wild type cells, and the newly captured mutant cells will be classified using these established ICGS states. And this allows you to make accurate comparisons to identify differential population changes in the mutant, as well as differential expression. Very powerful tool. So to analyze our R412X mutant mice, we captured granulocytic monocytic progenitors as well as CD11b positive cells for single-cell RNA-seq. Using cell harmony analysis, we were able to assign cell states to each of the newly captured mutant cells, shown by the black tick marks. Here first in the granulocytic monocytic progenitor cells. And we note that in the middle there, you can see a drastic expansion of monocytic cells and a contraction of granulocytic cell states. However, if we enrich for CD11b positive cells, we do find cells in the later stages of development, including IMNU in our mutant mice. If we focus on our newly captured mutant cells, we can see that the gene expression patterns of these mutant cells mimics that of the wild type, which I showed previously. So it appears as though granulopoiesis is intact in our R412X mutant mice, but it's severely dampened. Next, to understand the gene deregulation that is proximal to our GFI1 mutation, we focused our analysis on direct GFI1 target genes identified by CITE-Seq. And shown is differential gene expression versus wild type. Although there are a few genes that are deregulated in all of the cell states, and there are many more that exhibit broad deregulation patterns, there are a striking number of genes that exhibit cell state-specific deregulation, suggesting that a single mutation in a lineage-determining transcription factor, such as GFI1, differentially affects target gene expression in distinct cell states. So this highlights the power of single-cell RNA-seq and focusing your analysis on distinct cell states. And this will allow for more accurate identification of differential gene expression. To better understand the molecular mechanism underlying the gene deregulation seen in our R412X mutant mice, we hypothesized that chromatin accessibility could be altered. To address this, we performed single-cell ATAC-seq and performed label transfer from our transcriptional data. In the center, we focused our analysis on GFI1 target genes again. On the left plot, in wild-type cells, you can see either a progressive gain in chromatin accessibility throughout development or a progressive loss. However, on the right-hand side in mutant mice, this pattern is completely disrupted. And one of these deregulated loci is IRF8, which fails to close. Given the importance of IRF8 in antagonizing GFI1's function to promote granulopoiesis, we focused in on this locus. On the bottom, you can see that similar to wild-type GFI1, the R412X mutant can bind to the IRF8 locus. However, on the top, the chromatin fails to close in the mutant cells. These results suggest that the R412X mutant is defective in its ability to silence target genes. Given the noted deregulation in IRF8, we attempted to rescue our R412X mutant mice by performing an epistasis experiment. And although at the IRF8 het level, granulopoiesis is partially restored, those neutrophils remain abnormal. Additionally, if we breed the IRF8 GFP allele into our mutant mice, we find a significant deregulation of IRF8 in both GFI1 knockouts and our R412X mice. However, the homozygote mice have reduced levels of IRF8. This links both of these genetic rescues together and highlights the importance of repressing IRF8 to allow for granulopoiesis to proceed. However, repression of IRF8 and rescue of granulocytic specification is not sufficient to repair post-commitment defects in the neutrophils, as highlighted by the abnormal morphology. In summary, through our multiomics approach, we were able to demonstrate that a single mutation in a lineage-determining transcription factor has different effects on the expression of its target genes in each distinct cell state. We also highlighted the power of the cell harmony algorithm and its importance in making these conclusions. Importantly, our analysis identified two separate defects in granulopoiesis. The first is a problem with granulocytic specification due to deregulation of IRF8. However, there's a second post-commitment defect that cannot be rescued by lowering IRF8 allele dosage. We next turn to uncovering the cause of these post-commitment defects. To do so, we turned again to single-cell RNA-seq analysis. We performed additional captures of GFI1 heterozygous cells, K403R cells, which, as I remind you, are from mice with normal numbers of neutrophils. However, their neutrophils were functionally defective. And finally, of our homozygote or IRF8 het genetic rescues. You can see normal numbers of pro-new, pre-new, and im-new cells in the K403R mice, indicated by the green and yellow cells in the UMAP plot, which, similar to our FACS data, suggests that granulocytic specification and development is roughly intact in these mice. Additionally, you can see that the genetic rescues increase the incidence of these granulocytic cell types compared to the R412X het capture, illustrating a partial rescue of specification in these mice. Additionally, this is an expanded CITE-Seq analysis where we added in markers either identified in our data or from the literature. Shown here are the expression patterns in wild-type cells. Next, we calculated the differential expression of these surface markers versus GFI1 heterozygous cells. I hope you can appreciate that overall, the expression of these markers are not significantly changed throughout granulocytic development. However, we do see changes in expression of Ly6G in a few of the pre-new clusters. This again suggests that sorting solely on Ly6G is not sufficient to distinguish individual granulocytic clusters. Sorting solely based on the levels of Ly6G expression would lead to inaccurate comparisons between mutants and controls. Additionally, we see lower expression of CD106 or VCAM1 by ProNu2 cells in the R412X mutant. If you remember, VCAM1 is marking cells undergoing granulocytic specification, and the decreased expression of this marker is perhaps reflective of the specification defects seen in these mice. Notably, VCAM1 levels are partially restored in the genetic rescues. Overall, CITE-Seq has aided in improving our understanding of both normal and mutant granulopoiesis. We next focused on the impact of our genetic rescues on genes that were found to be deregulated in the R412X mice. Strikingly, we see a pattern similar to what we saw before. Those genes that are deregulated in each distinct cell state are also repaired in a cell state-specific manner. And these genes that are repaired are enriched in biological processes that are normally launched during the specification stages of granulopoiesis. However, a large number of genes remain deregulated even after genetic rescue, and these genes are enriched in biological processes that are normally launched during the commitment stages of granulopoiesis, and are enriched in functional processes such as the NADPH oxidase complex. This again suggests that repair of specification is not sufficient to repair post-commitment defects. We next sought to translate our findings to human, so we obtained neutrophils from neutropenic patients and performed single-cell RNA-seq. As shown in the middle panel, you can see that the genes deregulated in the neutropenic patients are enriched in genes that are differentially expressed during normal granulopoietic development in mice. And again, we see that these genes are enriched in processes involved in neutrophil function, as well as some involved in protein translation. Given this finding, it's possible that protein expression is deregulated in these mutant cells as well. So to investigate this, we isolated mature neutrophils from our mice and performed swath proteomics. We note a significant loss of neutrophil effector proteins, including many granule proteins and members of the NADPH oxidase complex. And this complex is responsible for forming reactive oxygen species to kill engulfed pathogens. So we tested the neutrophils isolated from our mutant mice for their ability to perform oxidative burst and found a significant defect. This extended to neutropenic patients as well. Our results identify an explanation for why neutropenic patients remain susceptible to recurrent infections. Our analyses reveal that the genetic rescue of neutrophil specification is not sufficient to repair post-commitment defects in neutrophil function, a finding that translates to human patients with SCN. In summary, our work exemplifies a multiomics workflow to understand the impact of human mutations on each distinct cell state. We began by performing functional assays to triage patient mutations and then modeled these mutations using novel mouse models of neutropenia. Next, we utilized single-cell RNA-seq and CITE-Seq to better understand normal terminal granulopoiesis before moving on to analysis of our mutant. This analysis allowed us to identify two separate defects, one in specification and one post-commitment. Further analysis using single-cell RNA-seq, ChIP-seq, and single-cell ATAC-seq allowed us to precisely determine that a single mutation in a lineage-determining transcription factor has differential effects on the expression of its target genes in each distinct cell state, highlighting the importance of analysis in separate cell states, as well as the importance of properly assigning mutant cells to their corresponding wild-type cell state. And finally, using an expanded CITE-Seq analysis, additional single-cell RNA-seq of primary human cells from neutropenic patients, followed by proteomics and a final functional assay, we determined that genetic rescue of specification was not sufficient to repair post-commitment defects in neutrophil function. It's our sincere hope that other groups will adopt a similar multiomics workflow to investigate the impact of other patient mutations in other systems. With that, I'd like to thank everyone who contributed to this work, especially everyone in the Grimes lab, and Lee himself for his excellent mentorship during my graduate work, for being a great scientist role model, and for the opportunity to work on this project. A special thanks to Andre Olson, who shared his expertise and performed the ChIP-seq and single-cell ATAC-seq studies, which were vital to our analyses. I would also like to thank Kyle Furchin, a current graduate student in Lee's lab, for helping out with the bioinformatic analyses. A big thanks to Nathan Salamonis for driving the bioinformatics analysis and developing the ICGS and cell harmony tools. Thanks to the Way Lab for help with our infectious challenges, the Let's Go Lab for help with iPSC work that I was unfortunately unable to share today, and the Grice Lab for running the proteomics. Kit Nazer at BioLegend was a big help getting us started on our CITE-Seq analyses. Finally, Cass, Cajun, and Jennifer provided vital patient samples and information, and Dietmar and Jayati for developing the GFI1 mutant mice, which we analyzed. And finally, I'll leave you with the title of our publication if you want to check that out and read into more detail. With that, I'd be happy to take any questions. Thank you for your attention.
Thanks, David, for that informative presentation. We have a number of audience questions already, so we're going to start the Q&A in a minute. First, I want to remind you that you can continue to submit questions using the Q&A box on your screen. I also want to let you know that we have added 2 resources to the resource section, an information sheet and an ebook, so you can check those out for more information. All right, David, are you ready to start the Q&A?
Yeah, looking forward to it.
Oh, good. All right, here's the first one. If neutropenic patients lack neutrophils, how did you analyze them? And did you analyze neutrophils from patients with GFI1 variants?
Oh, thanks for that question. So, although these neutropenic patients have extremely low neutrophil counts at the time of diagnosis, when they're diagnosed with severe congenital neutropenia, they are given doses of the cytokine GCSF, or granulocyte colony stimulating factor, and this pushes neutrophil production. And this is able to raise their neutrophil numbers and affords them with some immunity against pathogens. So they're able to go about their normal lives pretty well. So all of the samples that we analyzed were from patients that were on GCSF therapy, so we could isolate enough neutrophils for our analyses. Notably, neutrophils do not express many genes overall. As I showed in our data, we see a dramatic decrease in the total gene expression during differentiation. So for our single-cell RNA-seq analyses of the patient samples, we utilized the latest Fluidigm version 4 ultra-low input chemistry for library prep. And this allowed us for this to proceed with this analysis. Unfortunately, we were not able to obtain samples from patients with GFI1 variants. So SEN is already a very rare disease, and GFI1 variants have been found in only about a dozen or so patients. So most patients have Elaine variants, or Elaine encodes neutrophil elastase, and most of the samples we used in our study were from such patients. Interestingly, mice that express Elaine variants or are even Elaine knockouts have normal granulopoiesis. So there are some differences between mice and humans. So the mice that we generated with GFI1 variants are the first with knock-ins of patient mutations to exhibit neutropenia. But overall, it was really reassuring that our findings in the GFI1 mutant mice translated to human SCN patient samples from the patients that have variants in one of 3 different genes, including Elaine. So this suggests that our findings are broadly applicable in this disease.
Okay, thanks, David, for that explanation. The second question is, CITE-Seq and single-cell ATAC-Seq are powerful techniques. Can they be used in combination to analyze the same cells? And if so, is that what you did here?
Good question. So I agree that these 2 techniques are very powerful and each have their pros and cons. And while single-cell RNA-seq and single-cell ATAC-Seq can be performed in parallel on individual isolated nuclei, and this is to assess both nuclear RNA expression and chromatin accessibility. To my knowledge, there's not a current method that allows for simultaneous analysis of surface proteins by CITE-Seq as well as chromatin accessibility by ATAC-Seq. This will really be an extremely valuable tool if such a technique is developed. So in our studies, we performed the CITE-Seq and single-cell ATAC-Seq experiments separately, and we were able to compare the 2 data sets by performing what's called label transfer, which is a bioinformatics technique recently developed and published by the CITE lab. I also want to take this opportunity to mention that all of the data that we've generated here is reposited in Geo, but on top of that, Nathan and Stuart from his group developed a gene viewer where you can visualize the expression pattern of your favorite genes in our data. You can see their expression patterns in both normal and GFI1 mutant single-cell data. And so a link to a website that lets you access this viewer is listed in our methods section of the paper. Hopefully you can check that out. And I think that such tools are important to develop to make data from these multiomics data sets broadly applicable to biologists that do not necessarily have much bioinformatics experience. And really, I hope that in the future, more groups can adopt a similar approach to help make the data available to more scientists so we can make the most out of our work.
Okay, thank you. Before I ask the next question, I just want to remind the audience that you can continue to submit questions. We'll have time for them if you submit them soon. All right, the next question, David, is you mentioned that there are hundreds of total Seq-A antibodies available. Why did you use only 10 or so? Is there a limitation on the number of antibodies that can be analyzed simultaneously?
Good question. So for our very first experiment, BioLegend had only just launched their product line, And it was just lucky for us that the 4 markers were relevant to our analyses of granulopoiesis. And then later for our expanded analysis, the product line had expanded, but our goal was not really to perform an exhaustive CITE-Seq experiment using as many markers as we could, but rather we wanted to focus on the markers that were relevant to granulopoiesis. So we chose those markers that we found to be differentially expressed in our original data set, as well as adding in some markers that were published by other groups also studying granulopoiesis. And these markers were highlighted in their papers to mark different stages in granulocyte development. So theoretically, there's no limit to the number of total Seq antibodies you can use in a single experiment. I believe, if I remember, one group used a similar technique, or a technique similar to CITE-Seq, and published a data set with, I think it was around 100 markers. And I think there might be some technical considerations surrounding library prep and sequencing as the number of total Seq antibodies you use increases. But moving forward, I'm excited to see how other groups are able to use many, many markers in their analyses.
All right, David, thank you. Unfortunately, that's all the time we have. I want to thank you, David, for joining us today, and also offer a special thank you to BioLegend for sponsoring today's event. This webinar will be available on demand in about 24 hours. You can watch it again or share it with colleagues by visiting biocompare.com. You'll also get an email from us with links to the on-demand version. Thank you so much for joining us, and we look forward to seeing you at our next event.